Welcome to the second annual Sea Grant Coastal Communication Clips. This is a sort of a similar, I guess, rendition of the 3-minute thesis. We call it 3 minutes, 2 slides, and 1 message is the way we've been branding this. And I really want to thank the students that we have, I think, what, 16, 18, 15, 16 presentations, and so we're very excited. So the way the rules are is that I will basically start out with a title slide, and then each of the students will get up and give a 3-minute presentation. The focus of this is to engage you as a public. This is not, as we said in my email to them, I said, this is telling your parents and your friends what you do for your research. This is not at a scientific meeting. And so this is the art of communicating science, and I look forward to hearing each of them talk about their work.
Hi, I'm Nikki Button, and so I'll be talking about a new view of the Agulhas current today. The oceans are not static. Global ocean currents circulate the globe, as seen here in the bottom map. Western boundary currents, unfortunately some of the continents I guess are missing, can be seen flowing along the eastern coast of the continents. Most notably in the southern hemisphere is the Agulhas current, right here. The Agulhas current flows down the eastern boundary of the African continent and retroflects at this point here, flowing eastward. To note, Agulhas rings are formed and carry salt and heat into the Atlantic Ocean. Therefore, we want to understand this unique property of salinity data. However, until recently, a few years ago, we only had point data, such as from ships, Argo floats, buoys, or other similar data. Argo floats are tube-like devices that are deployed, and they take measurements of temperature, salinity at depth or at the surface. But with the launch of new satellites a few years ago, the SMOS soil moisture and ocean salinity satellite and the Aquarius satellite, we now are able to get a complete coverage of the entire oceans with salinity. So now we have this full data set, and you may wonder, well, what's the next step? Well, first we need to validate this data, because we cannot use it if we don't know the accuracy of it, especially to track these subtle changes in daily, weekly, monthly, or annual changes. So that is the first step of my research project. And then second, we want to be able to understand the influence of the Agulhas current on global climate change, especially because of this heat flow from the heat and salt flux into the Atlantic Ocean. So once we are able to validate that data, we can understand the role of the global climate, we can understand the role of the Agulhas current in the climate change and its influence on other currents. And so going forward, so it'll be very exciting to see how we can track these changes and monitoring this in this critical time period, especially with global climate change. Thanks.
So what happened to the ancient coastal Maya of southern Belize? My dissertation site, which is an ancient coastal Maya salt production workshop, is actually underwater in a peat bog in southern Belize. In 2013, I went into the field with the Underwater Maya project to answer several questions, some of which were, why is this site underwater? Was it once on platforms over top of the water? Or was it actually on dry land? So I chose to do this in several ways. But in particular, I chose to look at the one thing that could help me the most, the peat. The peat has preserved chiseled wooden posts, which acted as frameworks for the salt production workshops over 1,300 years ago. Of particular interest was a curved line of posts which ran along the edge of the site and looked like a retaining wall. So I decided how to look at this peat, what should I do? How could this peat help me determine the original location of this site? Well, if the site was once on dry land, then the peat should contain an abundance of plant material or organic matter. However, if the site was on posts over top of the water on platforms, then the organic content is going to be relatively low. So I took samples of the peat from the excavations and exported them under permit to the archaeology lab at LSU. I needed to find out two things. First, I needed to see how much plant material was actually present. And then I needed to figure out what it was. So I decided to take a small sample of the peat and analyze it under a microscope. There were hundreds of small roots, which belong to red mangrove trees. These trees keep pace with gradual sea level changes. So although I had found evidence that sea level rise had occurred, this still didn't prove the hypothesis that this site was once on dry land. I needed something else. I needed to figure out just how much organic matter was present, how much was in the sample. So using a technique which burns the sediment at over 400 degrees Celsius and turns that plant material into ash, I did just that. These values, in addition to the small red mangrove roots, proved one hypothesis and disproved another. The ancient Maya didn't build their site on a platform. They built it directly on land and then left when they could no longer outlast the rising seas. Thank you.
Hi, my PhD research focuses on understanding of the chemical contaminants found in the environment in different types of water with different salt content, like in an ocean versus a lake. But by fate, I mean when they are in the water, where do they go? Are they going to stay in the water, or they are going to partition into the air, sediment, tissue, and how much will go in each phase and how fast they are going to degrade? What tools we can use to answer this question? Like human, chemicals have fingerprints that differentiate them from each other, and we use those fingerprints to answer our questions. You may ask, why do we need to know the fate of chemicals in the environment? Let's answer that question with a famous example, DDT. DDT is a banned insecticide because the problem it causes in the environment. Like it is carcinogenic in human, or in some bird it causes eggshell thinning. What we know about this chemical is that when it is in the water, it doesn't stay there long. It will like to partition into the sediment or fatty tissue, and then it is very resistant to degradation. It can stay in the soil up to 30 years, or in the human body up to 6 years. And because it is banned, its levels are decreasing, but it is very slow. It took 20 years for DDT to get from this level to that in California sea lion. So the answer to the question, why we need to know the fate of chemicals, is that to make better decisions and protect our environment from the problems like DDT. These are some of the chemicals that I work with. This is herbicide atrazine, and these 4 chemicals are in the crude oil. As you can see, these 4 are very similar structure-wise, but the only difference that these have are these parts that we call them alkyl side chain. My research showed when they are in the water, for example after an oil spill, the larger the side chain, like this, the slower they are going to degrade and let stay in water, and they partition more into the air and fatty tissue. The first compound had shown health effect on fish and burial. It must ring a bell. We have to watch out for these other 2. Or for atrazine. If you put atrazine in the ocean, it will stay more in the water, but if you put it in the lake, it will partition to the fatty tissue more, so it can hurt a lake organism more than an ocean. So the take-home message is that chemicals with different structure behave differently in different types of water, and we need to consider that in our decision making.
Imagine that you are in a situation, blindfolded, standing middle of a road. The possibility of hit by a car is very high. You do not know from what direction the car will be hitting you or what could be the speed, how much injured you will be. In a nutshell, more specifically, how vulnerable you are in that specific situation. I'm putting you in this horrible situation because I want you to think about people who, a community of people like 400 million who live in the catchment area of the Bay of Bengal. They have exactly have the same situation but different scenarios. They face this at least 2 times in a year. Now, come to the real situation. Instead of the car, think about the cyclones or hurricanes. And instead of you, think of those 400 million peoples. Those people are vulnerable in terms of facing so many storm surges which can be 10 meter high. They are vulnerable because they face a deadly cyclones which cause lots of deaths in their life. So, in my thesis, I'll be focusing on the use of a statistical copula, which is a multivariate joint distribution method which will look for combined risk of wind speed and storm surge in that specific area. Copula removes the influence of the marginal values. So, from the distribution and the correlation value, we can see there is a strong correlation between wind and the storm surges. And copula lead us to study about this risk in combined together. As we see in the graph, as the wind increases, the storm surge increases. About 10% of the storm surge happen in the coastal, in the Bay of Bengal area. And among those percent, 1/6 has been the landfall in the coast of the Bay of Bengal. In my study, among 79 records, 53% has the landfall over the coast of the Bay of Bengal. Bay of Bengal has 40% of the landfalls among the whole Bay of Bengal's cyclone events. So, this study, which has been never done in this basin before, has a great significance to study for making the future coastal decisions. Thank you.
Okay, so by a show of hands, who here remembers the Slinky? Okay, great. Well, if you're unfamiliar, it's a toy that's really just a large spring, and it's targeted to entertain children who are easily amused. Some fun things you can do with it is watch it slink down the stairs or stretch at arm's length and let it snap back together. So what does this have to do with my graduate research? Well, the Slinky's ability to snap back into its original form is a great analogy for ecological resilience, which is the ability of an ecosystem to recover from a disturbance and return to its initial structural and functional levels. So for my thesis, I'm assessing the resilience of the Florida coastal Everglades mangroves to Hurricane Wilma, which was a Category 3 storm that crossed South Florida in 2005. I'm conducting my research at these 3 sites along Shark River, which are a part of a long-term ecological research project that started in 2001. This general trajectory of recovery from disturbance is the model I'm using to evaluate Hurricane Wilma's impact. And the recovery to pre-disturbance functional levels would suggest the system is resilient. So I want to relate the hurricane wind speeds to the disturbance magnitude or decrease in productivity and see how this interaction influences the recovery time of the mangrove forest at my 3 different sites. Or if I was studying slinkies, I would want to see how the varying degrees of stretching it would affect how quickly it comes back together, if at all. So here you can see the physical destruction from Hurricane Wilma. And these photos are of the same mangrove stand at the mouth of Shark River where hurricane wind speeds were highest. This severe canopy defoliation here indicates a high disturbance magnitude. And these damage and recovery photos are a visual representation of resilience. Some of my preliminary results show that 2 of my sites recovered from Hurricane Wilma within 5 years. And I was able to attribute the differential in recovery time to the varying wind speeds. So much like a slinky, these forests are able to bounce back. And this evaluation of resilience capacity of mangroves in disturbance-prone regions are important for understanding the implications to critical ecosystem services. Thanks.
Love hurts. Can you imagine you fall in love with someone in a bird shop, following him to a small coastal town, but being attacked by thousands of birds and almost lost your life? The Birds by Hitchcock is my favorite horror movie. He was inspired by hordes of dead birds washing up onto a coastal town in California. It turns out that bird attacks and their bird dying incidents were due to feeding on toxic algae produced by nutrient enrichment. Nutrient enrichment causes hypoxia, overly low oxygen concentration in water. This is the map of this year's hypoxic zone, and this is the goal we're trying to reach. As you can see, we still have a long way to go. Except nutrient enrichment, sea level rise caused by climate change is another main stressor for coastal rivers. Sea level rise will cause saltwater intrusion, which will change the distribution and transportation of nutrients, carbon, and metals. Intensive studies of nutrients, carbon, and metals have been done on the Mississippi and Atchafalaya rivers. However, little is known about other coastal rivers in southwest Louisiana. Therefore, my dissertation research will address the issues I just introduced with the focus on southwest Louisiana. These 4 rivers are used to study the inputs of nutrients and carbon to the Gulf of Mexico and their trends. The Calcasieu River is used to study how sea level rise impacts the distribution and transportation of nutrients, carbon, and the metals in a coastal river. Until now, we have sampled on the Calcasieu, Mermentau, and the Vermilion rivers monthly for more than 2 years, produced 3 papers, and 2 of which have already been published. The findings in my PhD study were fill a knowledge gap concerning the quantity and the characteristics of nutrient and carbon inputs to North America's largest hypoxic zone. And this is also the first assessment of the spatial and temporal variation of nutrients, carbon, and metals in a coastal river for such a long period of time. Thank you.
Okay, good morning. So today I'm going to talk about white spot syndrome virus in Louisiana crawfish. So as we all know, crawfish are culturally and economically important to the state of Louisiana. Crawfish farming or aquaculture is a multi-million-dollar industry every year and is the most profitable aquaculture industry in the state. Unfortunately for crawfish farmers, back in 2007, a shrimp virus originally from Asia called white spot syndrome virus made its way to Louisiana. This virus can cause high death rates in crawfish, and back in 2007, some farmers saw up to 90% dead crawfish in their traps. So for my master's research, I'm interested in learning more about how this virus infects and causes disease in Louisiana crawfish and the potential impacts it could have on the crawfish industry in the state. So in order to do this, We caught crawfish, wild crawfish, from the Atchafalaya River Basin that were naturally infected with white spot syndrome virus. We were able to remove the virus from the tissues of the animals, and this is a picture of what it looks like in the microscope. We were then able to perform studies where we took different amounts of the virus and infected crawfish in order to figure out how much of the virus you needed to cause disease and death in these animals. This is an example of one of my results charts showing that higher amounts of virus can kill 100% of crawfish, and lower amounts you get survivors and some animals that are not affected at all. So while this virus is definitely important for crawfish farmers, it could also have impacts for other industries in the state, such as shrimp. We found the virus in the Atchafalaya River Basin, and this basin spans a large area of the state and connects to many other water bodies, including the Gulf of Mexico. So, as I mentioned, this virus is originally from shrimp, and it can also cause high death rates in shrimp, just like in crawfish. So if this virus is able to make its way from the Atchafalaya Basin out into the Gulf, it could potentially impact shrimp. We do not know yet if this is a possibility, but we have some research partners who are currently interested in looking into this situation. And the hope is that together, their research and mine, we can get a full picture of how white spot syndrome virus can affect species in Louisiana. Thank you.
Hi everyone. As we know, the Mississippi River not only brings the water, but it also brings the sediment. Along with sediment, it brings the nutrients such as nitrogen, carbon, phosphorus. Not only that, but it also brings the pollutants such as PAH, heavy metals, and PCBs, which are really harmful to our health and are carcinogenic in nature. Over the time, those nutrients and the sediments are deposited in the sediments. But over the time, they leach out of the sediments and are transported back to the water column, which is known as the flux. Now, unfortunately, we don't know how much of that gets back onto the water column because we don't have the accurate measurement of the flux. We don't have the accurate measurement of the flux. But the flux is really important to understand the fate of those nutrients and the pollutants in the environment. So, just to give an idea of why the benthic flux is important, assuming a couple of grains of salt coming out with reference to the area of this podium, we end up having 12,000 tons of that particular contaminants or the nutrients per year, which is a large amount. So now we know the flux is important. The benthic flux is important. So traditionally the flux is measured using the benthic samples, but the benthic samples modify the in situ chemical and the physical condition of the chemical and the physical condition, which affects the flux, measured flux. So to get a better understanding and the better measurement, we are going with a new approach known as radium-thorium disequilibrium, which is using a radioactive radium and thorium naturally occurring in the sediments to calculate the flux. So we can calculate the flux using the thorium, using the radium. Then once we know the radium flux, we can calculate
everyone. So today I am going to take you all to the world of microbes in sediment that is still considered as a black box, as we know so little about these microbes' role in transformation of nutrients and metals in sediment. So for this particular study, we chose to understand the role of microbes in phosphorus mobilization, mainly because of two reasons. First, phosphorus is an essential element for life and is the building block, and second is we are running out of phosphorus. So it is very important for us to understand whether the soil microbes that is commonly found can actually affect the mobilization process. There has already been done a lot of studies on analyzing the chemical aspect. However, there are very few, in fact none for the coastal Louisiana, that specifically focuses on role of bacteria. So for the first time, realizing the need of the research, we analyzed the effect of bacteria that is commonly found in phosphorus mobilization process. So for this reason, what we did was we collected the sediment from different domains in Wax Lake Delta and incubated it for 12 days with and without bacteria, Sivanella putrefaciens, which is a very commonly found bacteria. So now going into the results section. In the first graph, the red color is to indicate the bacterial added condition and the green is to indicate the natural incubation. From this graph, we can clearly see that in the bacterial added condition, phosphorus release increased almost 3 times. And not just phosphorus, we can see that iron 2 is also getting released. But if we look at the second figure, we can clearly see that the change in phosphorus to iron ratio over time is not linear. The ratio is changing over time. So this indicates the very complex metabolic pathway of the bacteria, which we need to understand further, and my study is trying to focus on understanding more detail of this process. So with this, we can conclude that bacteria are indeed very important in driving the release mechanism of phosphorus, and not just in phosphorus, there is a need for studying this process in other nutrients and metals as well. So also these bacteria are sensitive to the salinity and pHs. So with the change in the climate change scenario, it will be very interesting to study this process further. Thank you.
Hi, I'm presenting my current research on how exposure to crude oil is going to affect multiple generations' reproduction, but also how it's going to affect future generations' resilience to exposure of low-dose chronic crude oil. So first thing I want to talk about is what's a generation? What is multiple generations? In my study, I'm working with 3 generations, and I want to think about it as grandparents, parents, children. And when we're exposing our fish, we're doing so right before they start creating waves of eggs in the spring and the fall. So what would happen to the second generation if their parents were exposed? And what happens to the third generation if their grandparents were exposed? And if we put the third generation in oil, are they going to have developed some sort of resistance? Are they going to be able to handle the stress of oil better than the generations before them? And in order to answer all these questions, we're going to be looking at the success of reproduction in each generation. So, oops. That's supposed to be a picture of me swabbing a fish, but that didn't work. Must have sent the wrong one. But first thing, the way to assess reproduction is to look. Reproduction is just a process of a lot of chemicals trying to make viable and healthy sperm and eggs. So the way you normally could test for reproduction being successful is to look at the fluctuation of reproductive hormones in a fish based on exposure history. Now, the way you normally have to do this is based on plasma. But our fish are so small, they're about like that big. In order to get any plasma, I have to kill the fish. So we wanted to try to find a different way to do it. And what that picture was supposed to show was me holding a fish, taking a sponge and swabbing the fish, taking off the mucus. And then when I take the mucus, I can then analyze that instead of the blood. And we're finding that this could actually work. If you look at the first graph, you can see the beginnings of a correlation curve between plasma and mucus in the blood. 11 ketotestosterone in the plasma and then in the mucus of adult male killifish. Then we decided to apply this method to an ecotoxicological study. So we took it for fish that were exposed and not exposed, and we see that there is actually a disruption in the levels of 11 kT based on their exposure regime. So what does this mean? Well, that means we can use this method that causes virtually no harm to the fish for fish that are small, even smaller than mine, or endangered species. Thank you.
Hello. PCDD and PCTF are commonly called dioxins. They are persistent organic pollutants. Once they are released into the environment, it takes a long time for them to break down. They enter into the air, they contaminate landfills, they enter the sediments of lakes, rivers, estuaries, and coastal area. After they enter the coastal aquatic environment, they bioaccumulate in marine and estuary organisms. Eventually, they might come to our dinner table through food chain. Dioxins are highly toxic, and they can cause cancer and many other health problems. Like this picture shows the previous Ukraine President Oshchenko before and after he was poisoned by dioxins. Dioxins are byproducts of many types of combustions, both natural and human sources. They are emitted from incineration, thermal power generation plants, automobiles, volcano eruptions, forest fires, and etc. Most of the dioxins emission from combustion concerns with transition metals such as iron and copper. Iron and copper exist in the fly ash from combustion. Fly ash is the smallest portion that could go into the air, and it's a complex mixture of metals and organic compounds. Transitional metals can assist the formation of certain radicals from organic waste, for example, like phenol and chlorophenol. Those radicals combine together to form dioxins. Our previous study has shown the individual metals on the effect of dioxin formation. So now we are interested in finding out whether there are synergetic effects between iron and copper for the formation of dioxins. So I built fly ash surrogate model with different ratios of iron and copper. We investigate combustion conditions on the formation of dioxins. We found higher yields of dioxins formed from bimetallic surrogate system, which showed synergetic effects between iron and copper. Also, we found high concentration of iron of oxygen contributes to low yields of dioxins. Through this study, we can adjust the combustor's combustion condition to reduce the formation and emission of dioxins. Thank you.
Coastal Louisiana is incredibly vulnerable to flooding. In the aftermath of flood events, there are often millions of dollars in damages and also lost economic opportunities. One way to reduce these damages is to not have them in the first place. And better preparing for this is a principle of hazard mitigation. The National Flood Insurance Program runs the Community Rating System, which is a program designed to help communities reduce their risk to flood damages. There are currently 18 different approved activities ranging from improving education, reducing flood damage, and improving mitigation and response systems to help reduce your flood risk. Participating communities receive a score between 10 and 1, with 1 being the highest. And based on this score, you receive a discount on your flood insurance premium, ranging between 5 and 45%. Currently, only about 5% of communities in the NFIP actually participate in the CRS, so there's a lot of room for improvement. This slide shows two of the best ways to reduce your damage after a storm or before it. The first one is elevation, getting all of your property off the ground. And the second one is open space preservation or land acquisition, which basically means after your first main event, you can't rebuild, and therefore there's basically no risk of anything happening because nothing's there anymore. So in my dissertation, I'm going to analyze the factors that help determine a CRS score. These factors include socioeconomic status, income, education, population, local government types, and the number of years in the CRS program. I'm going to apply a multivariate regression analysis to these factors for about 100 communities across the United States and see what is determining these scores. This slide shows a snapshot of the CRS classes of, first we have the 42 participating communities in Louisiana. As of 2014, these are their different classes. They range between class 9 to 6. And here, this is from 2006, but this is a graph of the national ones. And so as you can see, they're pretty similar, with most of the scores being right around 8. And so that's only a 10% discount, meaning there's still a lot of room for improvement. So with my research, I hope to gain a better understanding of what is influencing these scores and see what is driving community planners and policymakers to do certain hazard mitigating techniques. And hopefully the ultimate goal of this is to reduce flood risk, to increase the discount you receive on your flood insurance, and ultimately reduce actual damage and have less property loss. Thank you.
Hello everyone, my name is Clay Tucker, and I often get asked what I do for a living. And my answer is always, I study the effects of hurricanes on trees. And the most common response I get to that, which is also my favorite, is, well, they die. That's what happens to trees. And I know instantly that I've been given the golden opportunity to teach someone something. And it's true, trees do die during hurricanes, and I wouldn't recommend planting water oak or pecan in your backyard. Those are some of the more susceptible ones to hurricane winds. But there are some trees that are pretty resistant to hurricane forces. This is an image of my site in Grand Bay National Estuary and Research Reserve in coastal Mississippi. This is an area where pine savannas grow right next to salt marshes. This is a perfect place to study how hurricanes affect trees. And this picture is really powerful. One, because it shows this level of trees that grow higher going inland. The trees nearest to the coast are battered by the winds the most, and so they can't grow as tall. And the trees further inland are protected by those trees, so they can grow taller. Another way that we can see how hurricanes affect trees is by using the annual growth rings in those trees. The year following a hurricane, we see that the growth in the stem of the tree is much less than the previous years. This means that growth has been concentrated in the crown, and it's for repair after defoliation from the hurricane. And so this is important for two reasons. One, we can see when, by using really old trees, we can see when hurricanes occurred in the past. And two, we know that these coastal forests respond very significantly to an ecological disturbance. So in this way, we can increase our confidence of future hurricane occurrences by increasing our knowledge of the record, and we can better prepare for coastal restoration processes of these coastal forests in future. I hope you've enjoyed learning about hurricanes and trees as much as I have talking about them. Thank you.
Okay, most of us are familiar with the concept of food webs and simple trophic pyramids, which are basically a representation of energy flows in the food web. Now, the base of the pyramid and the food web is made up of primary producers, plants, algae, and phytoplankton, which basically harness the energy of the sun and make it available to the rest of the food web. Now, the coastal salt marsh is a complex food web which has a lot of different primary producers, both on land and in the water. And these in turn support all the different consumers we found in the marsh, like shrimp, oysters, and fish that inhabit all the different parts of the marsh. So we need to determine how energy flows from the bottom level primary producers up to different trophic levels, to all the different consumers. So take the seaside sparrow. How could we determine what source of primary production is most important to him? He doesn't feed on the grasses or the phytoplankton directly. He's at least one whole level removed from primary production. What we need is a unique chemical signature that we can measure that tells us, that can help us trace primary production up through the food web. Fortunately, for those of us who study stable isotopes, we know that there is indeed a unique chemical signature that we can detect in the tissues of plants that we can see move up to the tissues of insects and then see in the tissues of our sparrow. So to get a holistic view of the food web, we need to look at all the different signatures of all the different primary producers and recognize them in higher consumers. And so what is the value of characterizing this energy flow? So the 2010 Macondo oil spill really emphasized for us the importance of understanding trophic dynamics in the salt marsh. We can see that certain primary producers, like these marsh grasses, were heavily affected. We know that large numbers of insects died off, and these impacts and others may have ramifications for the rest of the food web. And the salt marsh really is a key ecosystem in the Gulf for both sparrows and humans, as it supports commercially and recreationally important fisheries. And so I'm hoping that by studying food web dynamics using stable isotopes, we can both determine past impacts of disturbances and help predict future impacts. Thank you.
Hello everyone. My research focuses on answering the simple question, how pollutants interact with soil. Now the reason this is important and why we're trying to answer this question is because as water level rises and the population increases, we get a decrease in the amount of land that we have for agricultural purposes. So it puts a lot of stress on our soil and we have to produce a lot of food. So farmers use pesticides to get that food, which is great. We get our food on our table, everyone gets to eat, everyone's happy. But the problem is we don't know how those pesticides interact with the soil and what happens once they're in the soil. Do they stay there? Do they absorb and stay for years? Or do they leach through and get into the water table? To understand this interaction, we must first look at what soil is made up of. So let's dig into it. It's made up of water, solid, and air. Now the solid section, which controls your absorption, is made up of inorganic, organic, and biological. Now organic, that's where the magic happens. That's where you get your interaction with your pesticides. That's going to control where your pesticide ends up. Now organic matter, or soil organic matter, or SOMS as it's called, is very complex. And its complexity is due to its composition. It's made up of lignin, carbohydrates, and fats. Now through studying SOMS, you get an idea of its model. And its behavior consists of a condensed phase and a loose phase. In the condensed phase, you have the center, right here, the lipids. And then on the loose phase surrounding it, you got your lignin and your cellulose. Now using this, we can create, using this model, we can create a synthetic soil that has the exact same characteristics as soil, as real world soil, but has none of the complexities. So we can easily study it in our labs. And that's exactly what we've done. We start off making this synthetic soil using a silicon particle, and then growing on top of it are lipids. And then on top of that, we got our lignin. And then on top of that, we get our cellulose. And by varying the different lignins that we put on there, we get different soils. We have 3 right now that we're working with. This right here is our 2-tiered system, and it shows the different lignin that we have there. Then by using different pesticides and interacting them and running absorption experiments, we can see how molecularly they interact. Then from there, we can create a model. This model can be used in the real world to predict how this pesticide we spray on the field is interacting with the soil. And in hopes we create a better pesticide that does exactly what we want in the soil and we get that food on our table. Thank you all for paying attention. I hope you have a great day.
Good morning. When New Orleans Saints won the Super Bowl championship in 2009, this was what I saw on television news. I was impressed how much Louisiana people love football. Also, the impact of hurricane on this state and the people who live here impressed themselves on my mind. Fortunately, over the last 10 years, the federal and state government have invested a huge amount of efforts, including money, to flood protection and coastal restoration. For example, we have a number of marsh creation and sediment diversion projects in the place to fight against the continuous wetland erosion. Budget is always a matter, but more importantly, before we go ahead to fix each individual problem, a big picture of nature's rule on sediment dynamics under different conditions is really in need. To better understand the effect of hurricanes on the sediment transport in southern Louisiana, we developed a fully coupled numerical modeling system for storm surge, waves, and sediment transport. Thanks to the power of nowadays supercomputer, a large-scale transport process can be simulated within a relatively short period of time. With this modeling capability, we are in the middle of drawing a roadmap for sediment transport from the continental shelf all the way to the coastal wetlands. Where we can go from here. Louisiana is making the new version of Coast Master Plan 2017. Our research will help the state agency evaluate the influence of hurricanes on the planned projects. And according to the feature of hurricane-induced redistribution of sediments, we will be able to choose different strategies at different locations. Therefore, we can help make sure the limited resources and tax dollars can be spent in a more efficient way. Thank you very much.